So, welcome to the first computational pathology interest group and lecture series. As I start, I just wanted to let you know that the sponsors for this meeting are Omnics, and the Omnics crowd is here. Hopefully we get a chance to meet and talk to them. And this event also happened because of the hospitality of Doctors Mike Bisich and Barb. So greatly appreciate that. And also the work behind the lines, Linda, Rob, and Janine, and the student helpers. Right? So why here? Why now? Right? And so what I wanted to say is that when I started thinking about computational pathology, which was about 2 years ago, and Lance, 3 years ago when Lance sort of joined the department, most of my collaborations happened because somebody came and knocked on the door. And after a while, you're happy with these collaborations, and you don't lift your head up, and then you realize that if you do, that there's actually lots of interest on campus. And the fact that Pittsburgh has a huge reputation for both the educational institutes that we have, and also for the clinical research at UPMC. So it made sense that a Department of Pathology, probably one of the largest here, should be able to use some of the computational power that's in and around. And when we went and spoke to Mike, he was a big supporter of this idea and said, I think we should do this. And just for fun's sake, I thought I'll put together at least some listing of all the diagnostic companies that I could think of that I have interacted with in the past. You also see that Omnics is already here, Prognostics, and I think Rebecca was going to be here. Nanovision, Yang Lu just walked in. This is her technology being turned into a company. And not just that, there's more interest with the Pittsburgh Health Data Alliance that was put together. And Mike leads one of the arms of that, which is a Center for Commercial Applications of Healthcare. And also Pittsburgh Life Sciences. So if you go to the list, it will tell you all the places, all the companies that they're actually on the roster, right? And so it seems like there's tons of interest in the diagnostic world, and the spirit is pretty collaborative. A good example of that is Mike and Barb actually hosting this event at their house, opening the whole place for us. And the fact that there are lots of educational programs as well, which are joined between the departments, between the universities, and it seems like there's a critical mass of people interested in doing more of this computational work in pathology. So we thought that just having this would be a fun beginning. And just want to very quickly plug in for two talks, upcoming talks. We were hoping to meet once a month. The next one will be from Rebecca and Prognostics. She'll be talking about Barrett's. And then the month after that will be Bob Nishikawa at the Department of Radiology. He'll talk about radiogenomics. And we have put together a website. If you have feedback on it, and if you have things that you want to see on the webpage, in addition to announcements, papers, and such, please let us know. The email addresses are there on the webpage. And there's one other outreach idea that we ran by Liron Pentanowitz, who is here, which would be that we could devote an entire issue, peer-reviewed obviously, focusing on computational pathology research in Pittsburgh. And then there could be a call out so that we could get other folks, friends from say Boston or Cleveland, to sort of contribute. And this is both to highlight the work that's done here and the interest that's out there. Okay. So before I sort of introduce the speaker, I just want to briefly touch upon the science that I think would be fun to talk about, which is with any event of this kind, you would like to know, what sort of definition one would subscribe to when you say computational pathology. And I'm going to try and keep it broad. Mike and colleagues actually had a few papers in the last 1 year stating out what could be the role of computational pathology and how one could go about thinking on this problem. And I was just going to say that on the science end of things, there are other ways also to state the same thing, which is that most of you here are interested in disease diagnosis and prognosis. And the other place where you would think about pathology, obviously, would be to understand disease biology, right? And there are enough problems in this space for each one of us to sort of work on. But I think there's a third angle that I think is worth talking about, briefly, of course, which is that visual perception. So in the end, with all this, you do have an expert, pathologist, looking at this data, and at least 20 to 30% of your cortex is directly related to visual processing. And if you combine vision with other modalities, which play a role with vision and action and such, there's at least 60% of your brain engaging in processing visual image data, right? So I think that's pretty impressive, and this has been an active line of work in computer vision, in visual psychophysics, and it's an extremely long history in visual perception. And why would you, again, why would you think about this is that's really the reason why you have headlines like this. How much can a woman trust the breast cancer biopsy? This is after the JAMA article. I think most of you have seen this, as to how much disagreement is there between the pathologists. Partly to do with how they process these images, and partly to do with the quality of the data. But the thing is that, of course, the fact is that this diagnosis is part science, part art, and that the biology is complex, and that somehow you need to integrate all this, keeping that visual perception in mind. And you also see in this other study wherein a room full of radiologists were asked to process images like this, and then a lot of them missed out that there was a gorilla in this picture. I thought most of you have seen this before. I'm not making a big deal of that. So given that in mind, which is that this diagnosis, prognosis, and this disease biology, how do we get this visual perception? How do we integrate this? And I feel like the colleagues we have here in Pittsburgh, I think most of you are actually attempting several of these problems. I thought that it would be fun to say what would be the 10 big ideas that would push this field further, right? And not surprisingly, you'll see that the speaker actually is engaged in many of these. But for fun, let me quickly take you through them. One would be, quantifying heterogeneity. The inter-tumor heterogeneity is well known, but the idea is that intra-tumor heterogeneity has a lot to say about tumor progression, has a lot to say about how you respond to therapy. How do you actually quantify that? How do you understand this spatial interaction between the cancer cells and the rest of the population in the tumor microenvironment? And I feel you already have seen many papers now in the literature talking about this in a big way. And the second thing would be wherein there's no reason for us to only think about hematoxylin eosin stained images. You could think of all the other transmitted light sort of ideas, which is the immunohistochemistry, or you could step into the biomarker world and think in terms of the fluorescence. Right? And I think an ideal way to think about these algorithms would be that you actually want to integrate information across all these channels because each one of them has something to say. I'm sure there's lots of redundancy because of the way the tissue is cut, and then tissues nearby are processed for hyperplexing or for other sort of biomarker experiments. So you need algorithms that will align them. You need algorithms to actually interpret them, and then the interpretation is sort of consistent. So if you take that to the next logical sort of step, you're really asking, not only are you worried about HND and the IHC or the immunofluorescence world, perhaps you also want to bring in all the genomics and the epigenomics data, right? So then you have a pretty unified view of the patient data. And it's not very surprising that there are data sets out there which allow you to do. This is the Allen Brain Institute data. It gives you the FISH data, it gives you the histopathology data, it gives you the RNA-seq data. Right? And the idea is that how do you integrate it? And a lot of people already here working. I think Kayan is interested in stuff like this. He has done this with MRI images. Takis is here. I'm working with Takis and doing stuff of this kind. So I think that would be big as long as the data sets get available and that we could actually use them. And so then sort of moving on away from biology and the disease biology and the disease diagnosis and prognostics, you could think about what it means to actually get the ground truth. As you know, most of these algorithms would work if only you had the ground truth. And it does seem like an old-fashioned way of doing it is that we asked Doug Hartman for data from Jan Lu's lab to actually load up those images on his iPhone and actually annotate it for us, right? You could imagine, well, that's just one person. What if you want more? And this is what Andy Beck and his group have done. They actually did a crowdsourcing of this annotation, wherein they can actually ask people to go and pick out the nuclei and such. And maybe you can do more. So in this case, we use tools again to annotate every pixel in the image. Right? So it's as if you're recognizing a scene and you would like to know what is going on at each pixel. But this could be tedious if only you had tools that we are putting together. A bunch of us got together to do this. If only you had tools which would automatically pick out all these interesting histological structures, and then if there is a diagnostic relevance attached to them, they could line them all up and say, hey, this is the most suspicious portion of the image or the data, and this is the least suspicious, and then put down a ranking, a risk ranking for that. So having said that, while it's very tempting to say that you need data, and I think there's no question about that, and you also need ground truth annotation, no question about that, it's very interesting to see what visual perception is doing. If I show you a cartoon map, and I meant if you're all pathologists here, that cartoon map is enough to communicate information about the tissue for you, right? And I think that's sort of an abstraction wherein you can just go from the tissue all the way to the cartoons and then communicate this information, meant that there was a lot of redundancy in these sort of data sets. And if only you could use that, if only you had a way to use that, then the issue about how much storage and all are, are non-existent. And in fact, much of the computer science, I think, is moving on those lines, wherein the key question is that what sort of statistics about these data sets that we could use to actually understand more? So can we have generative models for these data sets that allows us to abstract away the information in these images? And but if that is the case, and that you could actually get somebody interested in doing all this, perhaps you could also have some eye tracking sort of machinery, I'm going to call that as active vision, so that you actually understand what is the cognitive overload on the pathologist. How exactly do they think in making those decisions? And then how do you get a machine to actually learn some of those patterns so that maybe it's more aware of the errors that might possibly happen, right? And personalized medicine is a popular word, and I didn't know what to put here. You could make these machine learning algorithms be pretty good for a particular office or a particular pathologist, because maybe the training is different there, and if you get these machines to adapt, just like if you were to build a Google search sort of engine just for law documents, and that it would do something that's very different from what it can do for a medical documents, right? So I think that's all possible, and I feel like the end point is wherein show me an image, I can generate a histopathology report for you just by having the visual processing and also some language processing sort of built in. And indeed, it is the case that it should be able to give you spatial relationships. It should be able to tell you who is next to whom and such. And people often ask me, is this all real, or is it just that you came from computer science and you think you can do it? And that's the delay. So I'm going to try and play for you this video, and I'm sure you've seen this, which is that you don't need to drive cars anymore, right? So Google can take you around. And let me anyway. So the other one, without taking up too much time, the other one was a SpaceX landing recently in April 1st week, wherein the booster rocket comes back on a small floating ship down there, right? So I feel like the technology is out there, and in ways that is extremely interesting, and the fact that we have data that we can actually work with, we should be able to do this with the pathology. So with that in mind, I want to introduce Andy Beck, who is our guest speaker today. He's an associate professor. Should I swap the presentation? Yeah. He's an associate professor at Beth Israel Harvard Medical School in the Department of Pathology. in Beth, Israel. I told him I don't know how to pronounce deaconess, and I hope I got that right. He was at Brown for undergrad, did his residency and PhD in biomedical informatics at Stanford, then joined Harvard, and I think his work really captures the spirit of computational pathology, so it's an honor to have him. It's a pleasure.
Well, thanks so much for that introduction. I think that was a really great overview of the field, and thank you, Mike, so much for inviting me. This is a really exciting event to be a part of. And today I'll be talking about next generation computational pathology. And my disclosure is I'm a co-founder of a new startup called Path AI. And here's the big overview of my talk today. And I think it builds on a lot of what we saw in the introduction. What I think are some of the three key areas of the future development of computational pathology. The first is we can generate more and more complex data out of smaller and smaller tissue samples. And there's just been huge advances in molecular biology and bioengineering over the past few years. I'm going to talk about one of these applications that we're really excited about called expansion pathology. But I think overall, we all know that we're being able to generate more and more data sets from very small specimens. And pathologists are quite good and have been optimizing over the past 100 years to do really well on morphology or maybe single biomarker. But we're reaching a point where it's relatively cheap and easy to do multiple biomarkers on every single cell. And everyone knows that's a huge challenge, a huge cognitive load. And this sort of push for getting more and more data out of smaller and smaller specimens, I think, will be one thing that drives the further development of computational pathology, and to have it be more taken up both in research and clinical medicine. And I think something that may have hindered the uptake up till now was that the images that we were digitizing were essentially the same as the images that were being viewed under a microscope, whereas I think that's pretty rapidly changing, and I think the real onus is to prove the value of these methods, and that's one area we're really interested in. The second part of my talk is going to be about crowdsourcing. So the question is, once you've generated all of these huge data sets, that's not enough to do anything really of value and intelligent with, and you need to enrich the data set with human expertise to really label it for specific problems. I think there's going to be increasing need for more and more crowdsourcing or expert sourcing or just ways of getting very large, high-quality, well-annotated data sets. I think that's a major challenge for the field. It's becoming more and more easy to generate large data, but harder and harder to actually get this data in a format where it's useful for machine learning. And that's what I'm going to touch on in the final part of my talk is, what are the state-of-the-art machine learning algorithms? And if you do put together a well-annotated data set, what can these algorithms do? So the first part of my talk, I'm going to focus on this expansion pathology, where the goal really is to generate big data from tiny specimens. And this is a project we've been working on with Ed Boyden's group. Ed Boyden developed expansion microscopy and published the first paper on this about a year ago. And over the past year or so, we've been trying to optimize this method for use on totally routine pathology specimens. So really starting with the easiest possible specimen to actually get this involved in the clinical workflow, which would be a cover slip to H&E stained slide. And I want to briefly go through the overall molecular biology here let's see, actually this is the next slide. So you start with a cover slip slide, pre-stained. Maybe this is a challenging case that you want to extract a lot of additional molecular data on. So you can deparaffinize, decover slip, deparaffinize, perform antigen retrieval, immunostain for biomarkers of interest, anchor your biomolecules of interest to a gel, and then perform gelation to essentially have the tissue labeled, embedded in a polyacrylamide gel, treat aggressively with proteinase K, and then essentially that cuts all of the cross-links among proteins, and then expand it like a balloon with water. And you can essentially inflate tissue labeled with biomarkers of interest using water and expand it something like 5-fold in 3 dimensions. And this is if you do it once. You could also do this arbitrarily many cycles of this. So you can really blow tissue up very large. So one question you should be asking yourself is, why in the world would you want to do this? It sounds like a terrible idea. So that's what we've been working on, trying to figure out, is there any actual utility to this? Because it does seem pretty hard to believe that you could just blow up tissue with water and have it do something useful. So here's one example. This on the left is sort of an atypical introductal epithelial proliferation in the breast. There's a few atypical cells. This is pre-expansion, where you see this 20 micrometer scale bar. And we've essentially performed decover slipping, the whole procedure I described previously, on this tissue blew it up 5-fold, and you can see incredible preservation of the morphologic relationships on this image on the right, where you can really identify every single cell, except now it's 5-fold bigger. We've labeled it with 3 different biomarkers. And even from here, you can see that we have more resolution into what's going on with these individual atypical cells. And just to really show that in high power, here's one cell pre-expansion, where it's very difficult to make out morphologic detail. And then post-expansion on this one nucleus, we see very rich internuclear detail. We can start to see different patterns in the way the chromatin is clumping within multiple locations within a nucleus. We can start to see different patterns. This is a mitochondrial stain, the way the mitochondria is spreading out along the nuclear membrane. And I feel like these are all totally unexplored areas of disease pathology that now we have the tools to actually visualize these using conventional fluorescent microscope. And there might be a lot of biology we don't know that's important here, as well as a lot of new biomarkers to predict who's at risk or who isn't at risk. So I feel like this expansion microscopy really opens up a whole new set of different features that might actually be clinically useful, because it may turn out that if you have a nucleus that has this pattern of mitochondrial sort of layering along the nuclear membrane, that those are the cells that truly are preneoplastic and other ones aren't. And so we're really excited to try to explore these questions more in the future. So it becomes an incredible computational challenge once you've expanded tissue in 3 dimensions, as well as multiplex biomarkers on every single nucleus. So we've been focused in recent months on developing computational methods to identify, segment, and quantify features of nuclei from these 3-dimensional images. And then we wanted to see, are there any specific pathology questions that this can really help solve? So we had to think pretty hard, like, where is resolution actually important in pathology? Because the truth is, it's not important, at least we don't think it's important for many things. The simple answer to that is, most pathology is not done at 40x. Most pathology is done at 2x or 5x or 10x. So why do you really want to expand tissue 5-fold? Well, one area where that's not true is diagnosing medical kidney disease. So minimal change disease is a very common pediatric disease of the kidney, the most common cause of proteinuria in the pediatric population, and it requires electron microscopy to make the diagnosis. And the reason it requires that is because it's a disease of the podocyte foot processes, whereas here they're labeled in green. And you can see the reason it requires higher resolution than can be achieved with the standard microscope is here on the left you have this pre-expansion image, and you see this sort of flat area of green where you can't really see any tertiary foot processes. So you don't know if it's blunted, you don't know if it's normal. Whereas post-expansion, you can actually start to see little spaces between these tertiary foot processes, suggesting that this is a normal kidney and not involved by pathological processes causing blunting of the foot processes. So we were really excited to see this kind of lawn-like morphology post-expansion in a kidney not requiring electron microscopy. And then we asked the question, can you actually tell the difference between images of minimal change disease versus normal kidney post-expansion? And it looks like you can. So this is just one image where here we see the process of the tertiary foot processes, their morphology post-expansion in a normal kidney, and then we can see blunting of these tertiary foot processes in minimal change disease. And to explore this further, we just completed an experiment where we blinded a kidney pathologist to the diagnosis, and did show, in fact, that about 90% of the time, or actually 100% of the time for minimal change disease, not for one other disease, they could make the diagnosis on conventional microscopy in our experiment using expanded tissue, whereas they had absolutely no ability to diagnose minimal change disease on the pre-expanded tissue. So that's one application that I think is pretty exciting, but that's pretty specific. I think the more general use of this method might be in enabling much cleaner, much more highly multiplexed analyses within tissue where you're trying to analyze both genomic alterations as well as protein alterations. So here on the left we see a post-expanded tissue that's been stained for HER2, and we see no HER2 amplification, and we also see very, very little background. So you can really make out the nuclei well, and you can count even in this image pretty clearly 2 of the red HER2 signals in every cell showing no amplification. Whereas on the right, we have this post-expansion image where not only is it expanded, but since it's expanded, it's really diluted out the autofluorescence. So there's very little autofluorescence in this image, and we can see clear overexpression in every single cell of the HER2 protein, which is outlined in blue, as well as the HER2 DNA amplicon, which is labeled in red, as well as the green control probe. So this combination of having higher resolution morphology along with much higher resolution multiplex biomarker, I think could be really powerful for generating much more high quality data sets from standard pathology specimens. So to really test this in another more general scenario rather than diagnosing minimal change disease, we asked, could we build automated diagnostic models post-expansion, and would they perform any better than performing automated classification models on pre-expansion tissue? And we looked for within the setting of breast neoplasia, so starting from normal breast to ADH to DCIS to invasive breast cancer, and essentially had a set of images stained with H&E, as well as a set of images that underwent expansion and were stained with a nuclear stain. And we applied very similar procedure to each one, including image processing, feature extraction, construction of classification models, and then evaluation of classification performance. So one thing we saw early on was even for image processing, post-expansion helps in nuclear segmentation. It makes these low-level tasks more accurate by both reducing overlap between neighboring nuclei as well as reducing autofluorescence in the background. And then we evaluated how well did the models actually perform. And in each case, this is a relatively small experiment, so I think the actual estimates may or may not generalize extremely well to another data set. I think there's probably pretty big error bars around it because the data set's small. But I'm pretty convinced that expansion makes these models work better. So in every case where we build a classification model, we saw improved performance post-expansion compared to pre-expansion. And here we're doing things to try to build models to automatically do things like diagnose whether a breast biopsy was normal breast or usual ductal hyperplasia, normal breast or atypical ductal hyperplasia, and things like usual ductal hyperplasia versus atypical ductal hyperplasia, which is a clinically challenging area. For example, in the article cited previously from JAMA, there was almost like a coin flip in accurate diagnosis of atypia. And I think expansion plus multiplexing some biomarkers can really make this a much more solid diagnosis. So in summary, this is a new approach for physically expanding pathology specimens. It enables very high resolution analysis of morphology. It enables multiplexing in situ molecular assays with very little autofluorescence. And it's going to generate extremely large and complex 3D data sets from very tiny biopsy specimens. And it really turns into a huge data analysis challenge from this point on. So how are we going to deal with these huge data analysis challenges where now we can generate increasingly complex data with single cell resolution from very small tissue specimens, and they're far too complex for a single human without the aid of computation to deal with. So we already heard some of this from the first talk, and this is an area that I think is going to be extremely important, is obtaining expert or crowdsourced annotation of these large images. It's difficult to do this without crowdsourcing for a few reasons. One, because obtaining within image annotation from pathologists is difficult because they're not well reimbursed for it, they're very busy, they have large clinical workloads that have to take priority from this sort of work that's supportive of research. Pathologists are a very limited resource, their time is expensive, and these are two of the major reasons why there are no large-scale annotated pathology data sets that exist currently for machine learning algorithm development and evaluation. And as you said in the first talk, we now have really great algorithms, but they require data to be useful. And we really, in this field, are lacking high-quality data sets. And you could imagine any task where you got to really look carefully to find people who want to do it. And really the goal of the crowdsourcing is to find a subset of the world who's really going to enjoy and be good at doing things like labeling pathology images. So, those are two challenges that can be separated, and I think each problem can be dealt with. One is finding people, and the other is finding people who are really good at this, who are producing high-quality data that are actually going to make your algorithms better and not worse. So in terms of finding people, we used a platform called Crowdflower, which is a company in San Francisco that's essentially an alternative to Amazon Mechanical Turk. But they've, over the past 5 or 6 years, have built a very large network of workers all over the world who essentially go to their platform to find jobs that they want to work on, and then they get reimbursed for each job. So it's an avenue for accessing the workers around the world. And then the two tasks we initially asked them to do had to do with the low-level image processing task of nuclear detection as well as nuclear segmentation. So we trained them. These are examples of training images to either put a dot in the center of each nucleus and to segment each nucleus, which is a much more challenging task of drawing a border around each nucleus. We want to do this for a couple reasons. One is we want to use this to evaluate competing computational algorithms to get good scores for how well they're doing at both nucleus detection and nuclear segmentation. The second reason is we also want to use this data to train even better algorithms for nuclear detection and nuclear segmentation. And this is the sort of data that's produced from this. If we do this millions of times, then we can train, the world's best nuclear detectors and nuclear segmentation algorithms. So those are low-level tasks, but I think crowdsourcing is not only going to be useful for the very low-level tasks, we want to evaluate it for more and more high-level tasks. So the next sort of step up in terms of a high-level task would be interpretation of immunohistochemistry. And we've just completed a study in collaboration with the Nurses' Health Study where we wanted to ask how well can members of the crowd be trained to interpret standard immunohistochemistry assays like estrogen receptor and progesterone receptor. And this was a study on over 5,000 TMA cores and almost 2,000 patients. We tested two different forms of crowdsourcing. One form was having the crowd label each image to essentially say what the score was for the image in terms of expression of protein. And the other approach was actually to have the crowd label every single nucleus. So put a dot in every nucleus and say whether that nucleus was positive or negative. So as you might imagine, the nuclear labeling worked the best, but was considerably more expensive and took longer than the image labeling. And here's an example of the two interfaces they worked on. On the one hand, they were giving a score for the entire image, and on the other hand, they were giving a score for every single nucleus. And what the data showed was the crowdsourced nucleus labeling worked quite well, and this is with a diagnostic accuracy against a single pathologist reference of about 87%, which is very similar to the sort of inter-observer reproducibility you can see in scoring these biomarkers. The image labeling worked somewhat worse, and the actual worst performer in this was a commercial automated method that we applied. So I think this clearly shows that crowdsourcing is a real viable alternative to existing sort of turnkey commercial approaches, and also that this produces pretty high quality data that I think can be used to make these computational approaches even better. So the thing we're experimenting with now is beyond this very simple interface through Crowdflower of sort of asking people to answer questions, is this high positive or low positive, or is this nucleus positive or negative, to try to see if we can turn this into a game. And we're experimenting with a few different forms of game under a website we put up called Crowded Path. And this is still in development, but we have a few games that we think should be pretty fun where the user controls some sort of icon, and there's something like bad cancer cells falling into the bloodstream. You have to eat them before they get in the bloodstream. You have to avoid blood cells because you don't eat those. You get points for every cancer cell you eat, and then you have to classify a cell to get the maximum number of points. You have to correctly classify an immunohistochemistry image in this example for every cell you eat. We're also doing a sort of A/B experiment where we're also testing it against a sort of slot machine casino type interface where the goal is to earn as much money as possible. You earn money by doing well in the slot machine as well as by correctly classifying images. And of course, we only have a very small subset of the images labeled, so it's sort of like a random jolt of adrenaline or something. When you label an image that's been labeled already and you get it right, you get a bunch of bells and whistles. So we're kind of testing these, and ultimately we're going to test how much does gamification add, if anything, to just standard paying people for simple tasks. But I think ultimately for scaling this crowdsourcing stuff, doing these sorts of experiments should be useful. So in summary, we're leading a large-scale effort focused on figuring out how best to use crowdsourcing and gamification for computational pathology, because massive hand-annotated data really will be needed to fuel the development of improved next-generation computational pathology tools. And in the third part of my talk that I think is potentially the most interesting, I want to get into how are we actually going to use these large data sets we generate, which can be generated both from new advances in molecular biology as well as from large annotated data sets. And how can we use these expanded and deeply annotated data sets to fuel the use of increasingly deep and complex computational pathology models? And this portion of the talk is based on a project we completed about a month or so ago on building a deep learning-based cancer metastasis detection system. And this work was led by Dayoung Wang, who's a postdoctoral fellow in our group. And here the task was train the computer to automatically identify cancer in images of sentinel lymph nodes. The reason I think this is a good, simple, straightforward task to focus on for computational pathology is, one, it's an extremely clinically important area where evaluation of cancer in lymph nodes is needed for staging every single solid tumor and probably provides some of the most important information for determining prognosis and best treatment for that solid tumor. Number two, it's a very tedious and demanding task for pathologists. No pathologist, I think, enjoys looking through trays and trays of lymph nodes to find a few cancer cells that might be in the lymph node. And three, I think it's sort of intuitively a thing that computers should be good at because it requires very high concentration on very small regions of an image, and it's simply parallelizing that on a very large scale to cover a whole slide image. So the task here was, one, can the computer identify whether a slide contains cancer? That's the first task, the whole slide level prediction problem, which you could think of as a binary classification problem where essentially for each slide you're giving a yes, no answer. And the second task is a little more challenging. It's finding the metastatic location of every single region of tumor within a slide. And that's more of a segmentation problem where you essentially want to find each region and find the correct XY location. So in this experiment, we had about 270 training slides and about 130 held out evaluation slides. And this was done as part of the ISBI's 2016 Grand Challenge. So our basic framework, and a basic framework took by many of the teams, is pretty simple. So we have this labeled data set. It's about 2/3 of the data set that had been pre-labeled by a pathologist where the tumor, in this case, is outlined in green. So the green is the positive. Everything not within a green region is negative. We computationally extract millions of patches from either the regions of tumor or the regions of normal. And then we put these into a deep learning model. So the patch, the label, they go into an existing model. And then we train a model that can then take unlabeled patches, produce a prediction of whether that patch contains tumor. And essentially all of these patches that we make predictions on are pieced together to create a heat map that tells you the probability that every patch contains cancer. So the first task is extracting millions of patches from these training images, which is relatively straightforward, but I think an actually really important piece of the whole framework to do right. Because just like when you have a resident and you want to train them on the most informative examples, the exact same thing is true with deep learning. If you do this in a kind of knee-jerk way, it tends to not work quite as well as figuring out what patches the computer would learn the most from and enriching your training set for those patches. I think potentially the easiest part of the whole framework is actually selecting the network architecture, simply because so many companies over the past 5 years and academic groups have worked really hard to build really high quality deep learning architectures, and most of these have been open sourced and are publicly available. So in our experiment, we looked at 4 very high quality open source deep learning architectures, including GoogleNet, which won the ImageNet Large Scale Computer Vision Challenge in 2014, the VGG16 net, a FaceNet, which was something developed for face detection, as well as the original AlexNet, which broke a lot of records back in 2012 by doing considerably better than all previous computer vision approaches at the ImageNet Challenge. And basically we did quite well with all of these. So this is the patch level classification, which you'll remember is not one of the actual metrics we're using. We're using metrics at the slide level or the region level. But basically with GoogleNet, on held out cases, we were getting like 98.4% accuracy, which was reassuring, and that's why we ultimately went with that architecture. So brief overview of the GoogleNet architecture. It has 27 layers in total, a total of 6 million parameters with 3 loss layers. And it's described in much more depth in this paper produced by Google Research called Going Deeper with Convolutions. So what do we do with this network? Well, we take new images, break it into patches, put the patches through the deep network, and then we create this heat map, which we use to visualize the output of the network, where red indicates hotspot for cancer, everything else indicates predicted no cancer. So here's where we put a fair amount of our attention in this project, was figuring out how to go from this set of heat maps to an actual prediction at the slide level. because here we still don't know what to predict. So we built another model on top of our initial deep model, which was a model that took as input these heat maps and produced as output prediction tumor versus normal. And here we actually used a lot of our human intuition and sort of pathology expertise to figure out what do we think are the characteristics of the heat map that are more likely to predict whether you have tumor. And these were simple things that we sort of hand engineered, like percentage of tumor region over the whole tissue region, the area ratio between tumor region and the minimum surrounding convex region, under the intuition that regions of tumor would be more heterogeneous than regions of normal lymph node, as well as simple things like average prediction value over the slide, as well as the longest axis of the tumor region. So we essentially take these heat maps, we know whether on the training set whether it's cancer or not, we extract a small set of features and build a decision tree to then actually get the final diagnosis.
Excuse me, before you go further, so what is actually your input data here?
So the input data here is the heat map, and
You started from a human biopsy material.
yes, so we start with sentinel lymph node. Sorry, I'll go through the slides slower. So we start with a sentinel lymph node biopsy, and for each patch in it, we know whether it's cancer or normal, because a pathologist hand labeled cancer. So essentially we can break this into millions of patches. And on our training set, we know whether each patch is from cancer or normal. So then we can build a model that essentially does a really good job of separating patches into cancer or normal.
So you didn't just put tiles on it, right? Did you just put tiles on it?
You could, yeah. So we didn't exhaustively sample tiles, but I mean you could, and you'd probably get similar results. It would just be very computationally demanding if you exhaustively tile. But essentially the procedure is generate a bunch of tiles, millions of tiles. So essentially you have two columns, the JPEG name of your small tile and is it cancer or not. And you might have 3 million rows. And that's the input to this model, is a big set of images and a big set of labels. And then the output is we then build this model and fix the model that does best at taking as input a tile, producing a prediction of what it is. And then we apply it to new images where it doesn't know the right answer. And then produces a heat map, because the output of the model is for each tile a number between 0 and 1, where 1 means it's cancer, 0 means it's not cancer. And here we're representing that number with a color, where red means cancer, blue means no cancer. Any other questions? So the challenge here, though, and this might be kind of a subtle point, but I think it's an important point, is then how do you make the classification like that you're going to put in the pathology report? You don't just want to give a pretty picture. You want to say, is this slide have cancer or does this slide not have cancer? And here's where I think, I think this is sort of the future of deep learning, is integrating these sort of simple knee-jerk methods with how do you actually make sense of it. And what we did here was, here the input data were these heat maps, which is the output of the first model, plus whether the slide has cancer or not. So before it was patch level, does the patch have cancer or not? Here's does the slide have cancer or not? And then we engineered some features about this heat map, like how much red is in it, what is the shape of the red, how much more red than blue, etc. And then we built a model to predict the image level. So first we're predicting the patch level, here we're predicting the image level. And then we could actually look inside our model and say which characteristics of the heat maps are most predictive of cancer. And it was things like you might expect, like what is the longest region of tumor in the image, what are the ratio of pixels in tumor to ratio of pixels not in, the ratio of pixels in tumor to pixels in not predicted to tumor, etc. And then for the detection task, it was also pretty straightforward. We essentially took our heat maps, identified connected regions within it, identified the central region within each connected region, and then output the XY location of each central point within the region of the tumor. So essentially this one's very straightforward. It's just breaking this down into regions and outputting the position of each region. And the last thing we did was to estimate a very precise probability of tumor within each region we identified. We integrated 2 different models, which we thought would give a more robust estimate of the probability of tumor at every location. And that's all the math kind of and network architecture behind it. I want to give some images to show how these things actually work. So here's an image on the left. Do people think positive or negative? Any guesses from the crowd? Positive. Yeah, that's a pretty good positive ones. So all these big white regions are positive. This is an easy case, but the model did a nice job of correctly identifying the positive regions in red. How about this one? Uh-oh, the title gives it away. Don't read the title. Okay, so that one is hard. You might want to stare at this one for 20 minutes because there's millions or billions of cells in there, I don't know. But if you have this heat map, you can quickly focus on the ones the computer's worried about where it's bright red, and then go high magnification. And this is real held out data. Here you see like a very thin rim on the edge of this lymph node of actual metastatic tumor cells. Here's another area of red where you see this very tiny collection of maybe 10 metastatic cancer cells within this huge lymph node. So this could speed up interpretation of this case, 20-fold, because rather than staring at every single little thing, you can immediately focus on these red regions and see the tumor. So I briefly want to describe the actual results of this competition. These slides come from Beck, who did an amazing job, I think, of organizing this competition. It was one of the first competitions to take a large number of whole slide images. So this is much closer to the clinic than many of the previous competitions that had hand curated, extracted small pictures of things like mitoses. This was actual from several different institutions, like clinical whole slide images. And there are about 198 registrants from countries around the world. The countries that submitted the most were America, India, and China. And it was a pretty even mix between companies as well as academic institutions. Ours was the Harvard, MIT contribution. And it was interesting to see where the field is. So there are 23 final submissions. Even though almost 2 people registered and downloaded data, at the end of the competition, only 23 teams submitted. And about 16 of them used about 16 of them used deep learning methods. So 2/3 of the enters were deep learning, about 1/3 were classical machine learning. Classical systems are things like texture and intensity-based features that were then put into things like random forest, support vector machine, AdaBoost. Some of them did analysis at multiple scales. And some had sort of cascades of these kind of simple linear models, whereas 2/3 of the teams did deep learning, including leading architectures like GoogleNet, VGGNet, ResNet. So basically, as we all know, computer vision is using a lot of deep learning. And here's the output from leaderboard 1, which is the whole slide image classification. So here, basically, you take as input a slide, you produce as output yes or no for that slide. So the worst teams did a little bit better than random with an accuracy of like 56%. And then all the top 5 teams were deep learning, and our team barely edged out this company, EXV, in Germany, getting an AUC of 0.925 for this first task.
Who was the independent participant?
I heard it was one guy, but he worked for Roche. That's what
Okay.
But I don't know if that's true.
Sorry, if I understood correctly, so all the first 10 teams use deep learning, whereas the latter ones use the classical machine learning method?
Pretty much, yeah. The top 5, I know the competition people said the top 5, and you could actually read every single one of these, what they did on the website. But yeah, the top 5 were all deep learning, and the bottom ones were mostly not. So overall 2/3 of them used deep learning, but the top 5 were all deep learning. And then for the second one,
my question is, since these teams, the first 5 teams use deep learning technologies, but why they have some difference in the performance?
No, and that's
I think the deep learning technology itself is the same, right?
That's a great question. And yeah, the top 5 use deep learning, but also a lot of the bottom 10 use deep learning. I mean, so 2/3 of them use deep learning. So for example, GoogleNet, which we used, we were number 1. There was a team that was number 10 and a team that was number 18. So it goes to show you that this is the easy part. Picking the model you're going to use is the easy part. There's many challenging parts that actually make the difference from doing stuff well and not doing it not as well. I mean, in our case, I think what we did well was intelligently picking training patches has a huge impact on performance. And then the post-processing was enormously important. And that was just for this one competition, but I think in real life, yeah, I mean, picking the model architecture is, maybe one important piece. I actually don't even think it's an important piece. Once you decide you're doing deep learning, I mean, all of these things work pretty well. So I think it's really understanding the problem and thinking about how to do training better, how to do post-processing better. So for the second one, what these numbers mean is this is essentially the sensitivity, which means which is the proportion of cancer in a slide that you're identifying correctly with only about one false positive. So essentially the team that's last is only finding 10% of cancer with only one false positive. So this is a harder task because of this, rule that you can't have more than one false positive. And this one, we also won. I think we won by a considerably larger margin than the previous one. The previous one was tiny. Essentially, all the top teams did really well. This one, our team separated from the others. I think we did a couple kind of silly things in a better way than other teams that made a big impact on our performance. But again, the team from the EXP company from Germany did quite well, and the Netherlands team did as well. So just to give you some examples of how well these systems are working, here on the top of the image, the color, if it's green, it means that that person got it right. If it's red, it means they got it wrong. We have the pathologist on the left, and then the top 5 teams on the right. So like this is a tiny set of cancer that the pathologist and 2 of the teams got, 3 of the teams didn't get. This is probably one of the hardest ones, where this is cancer and like that's not cancer. So it's extremely visually subtle. And this one, this one our team got, and even the pathologist didn't get this one. So this is all kind of suggesting that these computational pathology methods, even today, have the ability to add information to what the pathologist is doing. Here's the ROC curves for the second task. And the red dot represents the pathologist. So the pathologist is essentially getting a 73% sensitivity with 0 false positives. And our system is getting a 73% sensitivity with about 1.3 false positives. So we think this can continue to get better, and even today it's approaching human performance. And then we asked, if you combine human with deep learning, do you get a better system? And it turns out you definitely do. So the pathologist in this case had an AUC of 0.9664. Our system had an AUC of 0.9250, indicated in the blue line. But when you combine the two, you get a huge bump in performance. So the pathologist goes from 0.9664 to 0.9948, which at least it's huge for this, where the pathologist is pretty good. So it's actually like an 85% reduction in error rate. And if this were distributed widely, and there's something like 4,000 sentinel lymph nodes evaluated per day, you could reduce daily errors from something like 140 to 22, and we hope it makes people faster and more accurate. So in summary, I think this competition shows promising performance for computational pathology for this simple task of identifying cancer in lymph nodes. I think there's pretty clear data that these systems will make pathologists significantly faster and more accurate. And I think the most exciting part is these deep models should continue to improve as training data becomes larger and richer over time. And in my last slide, I just wanted to briefly summarize what we discussed, as well as kind of discuss what I see as so exciting about the future directions of this field. So one is there's continuing to be huge advances in how we can extract huge data from very small tissue specimens, and one example of a method that can do that is expansion pathology. There's a lot of infrastructure out there now, and many companies have built nice platforms for using crowdsourcing, which I think can really help build more robust computational pathology algorithms. And I think the integration of these very large data sets with lots of annotation can drive the intelligent use of advanced artificial intelligence for really building high-quality, robust, next-generation computational pathology algorithms to make pathologists faster and more accurate. And then, as we all know, pathology is really central to a lot of research as well as biomedicine, and these improved tools can have huge impact on both research and clinical practice by impacting fields like molecular and cellular biology, systems biology, computer science, and clinical medicine. And I want to thank everyone for their attention, and I want to thank Mike and all the organizers again for inviting me. I also want to acknowledge my lab at Beth Israel, our collaborators with the Nurses Health Study, the organizers of the 2016 ISBI Camelyon-2016 Grand Challenge, our collaboration with Ed Boyd and Youngsing Zhao at MIT on the expansion pathology project, as well as the support our lab has had. And I look forward to answering any questions as well as discussing more this afternoon. So thank you.
So you had more than 3.5
also slide training and testing. So how the color variation among these slides, do you have to deal with that at all?
Yeah, so we did very simple color normalization. So there were two different institutions. They actually did have different color, and all we did was subtract out the mean in each channel. So we just centered. We didn't actually do any color normalization. And I don't know if we would have done even better, but we didn't even test that. And some of the teams, a lot of the teams did use color normalization. But I think these deep models, they're pretty good as long as you have good variety, like both cancer and normal from multiple institutions. Of course, if all the cancer came from one place, all the normal came from another place, and the color had artifacts, you would learn that artifact. But assuming you have sort of equal cancer, normal, and both, the model should be able to overcome it, I think.
Yeah, what kind of more high-level question? What would you say? Like, what part of the pipeline would you say requires the most pathology domain expertise? Like the feature generation or post-processing, or like where do you need?
Yeah. So I think first off, there's sort of the intricacies of a contest, the constraints, the artificial constraints of a contest. But in real life, I think it's within image. If you want the computer to do stuff that the pathologist is doing, like identifying cancer within lymph nodes, identifying mitotic figures, quantitating amount of tumor, then you need within image annotation. And that's by far the most important thing, is just labeling within image. I think the post-processing was important for the competition. I'm not sure it's going to be important if you have large enough data sets.
Danny, I have a question for you. This is just your prediction. Is how many years do you think it will take until these computational tools will replace the pathologists?
Well, first off, I thought you were going to ask how long until people are using them.
But I think
I don't think they'll ever replace completely, but they certainly might replace some pathology. I mean, that's something to think of. I think that's a possibility. Or pathologists will do other things. So I mean, the other way to frame it is it will.
It's okay I think this crowd are happy. They're okay. No one's going to throw anything at you.
Yeah. No. So how long until that's possible? I think someone needs to think of a very focused area where that could happen. It won't happen with lymph nodes, right? Because that's a very minority of pathologist practice. So you're not going to replace a pathologist because you have this lymph node thing. But if there is some task that is 100% of what someone's doing a day, and you get really good at that task on the computer, you might be able to, do the work of 5 pathologists, 2 pathologists. And how long do I think that'll take? I don't know, 5 years? I don't know. In one area. For example, there's already CytoPath. I mean, maybe they've replaced some screeners with the system. And I think it'll be something similar to that, a really well-defined question. And but when will they be using them? Yeah, I don't know that either. But.
I think the beauty of deep learning is that it can generalize to specific problems. So my question is, when you're feeding it the patches and the binary classification, do you actually have to tune any of the internal parameters of the neural network, or do you just give it that and then it's just an input-output system where it gives you
Yeah. So the cool thing here was it was just input-output. So we did not touch that virtually at all. And we didn't use a pre-trained model. So we trained it from scratch on all of our patches. And I feel like the heavy lifting was done by Google in terms of getting that thing to actually converge. And, building from scratch a 27-layer network is really hard. And yeah.
Also, you explained the expanded image.
Yeah.
Give more details. But my question is, this kind of expansion will cause the change of the morphology.
Yeah, so it doesn't really. I mean, there's some distortion, maybe less than 5%, but we also, I have a couple answers. It actually turns out it doesn't. That was this initial expansion microscopy paper was in Science, and they did very careful distortion tests to prove that it was less than 5% distortion. The other thing, though, is maybe distortion is not such a bad thing, because our goal is not to have the perfect readout of the underlying structure. Our goal is to do something that means something for patients. So you can actually imagine a scenario where if you're distorted a lot, that actually tells you something about the function of that nucleus. Like if you have a loss of some nuclear integrity, you expand more. And I think we need to just capture these data sets and perform those analyses to figure it out. But I don't think there's a lot of distortion. I actually don't think that's a huge problem for many applications.
So I think it counteracts the problem we have now, because we do the opposite. We shrink everything in formalin. There's 10% shrinkage, so we actually make it worse than the real state. So you're probably compensating for some of the artifacts that we deal with now.
Mm-hmm.
What about like in this expansion technique? What about like epitope plus for proteins?
Yeah, so the staining should be done before the expansion and before the aggressive protease treatment. Because yeah, you're right, that will mess up epitopes. Treating with the protease to cut all the inter-protein connections prior to expansion. So we do all the staining up front.
So you said you fed it about 3 million patches with the ground truth of 1 or 0. So did you derive that from the annotations, or did you actually have a pathologist go through the individual patches?
No, thank goodness. Yeah, so in this case, this is all part of the competition, what I showed today. These were whole slide images, and they said it took about an hour per image. They had multiple pathologists, but circle regions.
Okay.
So a circle might contain, 10,000 patches, but it would just be one circle. Because the other way of doing it is way harder, I think, to have people classify each individual patch.
Maybe I can have the last question.
I was wondering, how do you decide on the patch sizes? Do you change?
No, we just picked one and stuck with it. No, we didn't. I don't think other teams discussed this. I don't think it would affect performance much. But it just looked biologically. I mean, we did decide, but we didn't have any data-driven decision. It was just like, this looks informative. So basically, you want to show, and how do we pick the resolution also? So we did it 4DX. But you basically want to show pathologists or people who know what they're looking at, what's the image where you can best separate normal from cancer, and then go with that size and resolution.
I know that some computer scientists aren't particularly fond of like deep learning versus conventional machine learning, so I want to know if you can tell us how or why you decided to go with deep learning.
Because it works. I mean, I was trained in all different methods. I'm not like it was mainly because it's working better. I mean, it's as simple as that.
If we flip the question around and ask, what is it telling you about the data at the end of all this? What will you say to a layperson? I know you're a pathologist.
Yeah, so we haven't done that, and we could, this sort of like visualization of what the network is actually learning. I think it's an interesting question. This is such a practical question that I don't think it's necessarily going to be anything very interesting, right? Like cancer versus no cancer. So it's probably finding nuclei and probably separating on texture, but we don't know that. And I think at least for this very specific, totally operational type application where you just wanted to say cancer, no cancer, I don't know why it's saying it, but I think in a lot of models you wouldn't really know why very clearly. I mean, this other stuff people are doing is like sift features and texture features and throw them into an SVM. It's not like those are a whole lot easier to explain than this. So I basically don't really know, but yeah.
So with that, thanks.